For more than a century, scientists and medical researchers have been captivated by the mysterious plaques and tangles that characterize Alzheimer's disease. These abnormal protein deposits have dominated our understanding of neurodegeneration and driven the vast majority of drug development efforts aimed at combating this devastating condition. However, a groundbreaking body of research emerging from Purdue University is challenging this long-held paradigm and suggesting that we may have been overlooking a critical piece of the Alzheimer's puzzle hiding in plain sight: fat accumulation within the brain's immune cells. Leading this revolutionary work is Gaurav Chopra, the James Tarpo Jr., and Margaret Tarpo Professor of Chemistry and Computer Science at Purdue University, whose team has recently published findings in the prestigious journal Immunity that fundamentally reshape our understanding of how Alzheimer's disease progresses at the cellular level. Rather than focusing exclusively on the protein aggregates that have captivated researchers for decades, Chopra's laboratory has turned its attention to the abnormally lipid-rich cells that surround diseased regions of the brain, uncovering a complex metabolic dysfunction that may be driving the disease process itself. A historical perspective on an overlooked discovery. The story of lipids in Alzheimer's disease is not entirely new, though their significance has only recently begun to be appreciated. More than 100 years ago, when the German psychiatrist and neuropathologist Alois Alzheimer first examined the brain tissue of a woman suffering from profound memory loss and cognitive decline, he meticulously documented several key abnormalities. Among his observations were the now famous amyloid beta plaques and tau protein tangles that have become synonymous with the disease. However, Alzheimer also noted something else during his microscopic examinations: cells throughout the diseased brain that were filled with droplets of fatty compounds known as lipids. For decades, these lipid-laden cells were largely dismissed by the scientific community as mere products or inconsequential debris resulting from the disease process. The prevailing assumption was that these fat deposits represented nothing more than cellular garbage, the detritus of dying neurons, and degenerating tissue. This dismissal meant that lipid biology in neurodegenerative disease remained largely unexplored, with research efforts and pharmaceutical development concentrated almost exclusively on finding ways to remove or prevent the formation of protein plaques and tangles. The emerging lipid model of neurodegeneration. Chopra and his collaborators are now systematically dismantling this assumption through a series of interconnected studies that reveal lipid metabolism as central to brain health and disease. Their research builds upon earlier work published in the journal Nature, where they demonstrated that astrocytes, which are support cells that nourish and protect neurons, release toxic fatty acids in the presence of disease conditions. Additional collaborative research with the University of Pennsylvania, also published in Nature, established links between mitochondrial dysfunction in neurons and fat deposits accumulating in glial cells during the aging process, which represents the single largest risk factor for developing Alzheimer's disease. These findings have led Chopra to propose what he terms a new lipid model of neurodegeneration, a framework that places lipid metabolism and fat accumulation at the center of our understanding of how brain diseases develop and progress. According to this model, it is not the individual lipid droplets themselves that are inherently pathogenic, but rather their progressive accumulation within brain cells that creates problems. Chopra has coined the term lipid plaques to describe these accumulations, noting that they do not resemble the spherical droplets typically associated with fat storage, but instead form irregular plaque-like structures within cells. In Chopra's view, the specific composition of lipid molecules that accumulate within different types of brain cells may be one of the major drivers of neuroinflammation, the chronic inflammatory state that characterizes Alzheimer's disease and other neurodegenerative conditions. Furthermore, he suggests that the particular composition of these lipid plaques may actually define specific brain diseases, potentially explaining why different neurodegenerative disorders produce distinct clinical symptoms and pathological features despite sharing some common underlying mechanisms. The critical role of microglia in brain health. To understand the significance of Chopra's latest findings, it is essential to appreciate the crucial role that microglia play in maintaining brain health. These specialized cells serve as the brain's resident immune system, constantly surveying their environment for threats and performing essential housekeeping functions. Microglia are responsible for clearing away cellular debris, patrolling for infectious agents, and most importantly for Alzheimer's disease, removing misfolded proteins like amyloid beta and tau before they can accumulate into larger, more problematic deposits. Under normal circumstances, microglia identify these protein aggregates and eliminate them through a process called phagocytosis, essentially engulfing and digesting the problematic proteins. This clearance mechanism represents one of the brain's primary defenses against the development of Alzheimer's disease. However, Chopra's research reveals that in the presence of amyloid beta plaques, microglia themselves become compromised, setting up a vicious cycle that accelerates disease progression. Unveiling the mechanism of microglial dysfunction. The research team's investigation began with a deceptively simple question: what happens to microglia when they encounter amyloid beta? To answer this question, they examined brain tissue samples from individuals who had died with Alzheimer's disease, carefully analyzing the spatial relationship between amyloid beta plaques and the surrounding microglial cells. Their observations revealed a striking pattern. Microglia located within 10 micrometers of amyloid beta plaques, essentially in direct contact with these protein deposits, contained approximately twice as many lipid droplets compared to microglia positioned farther away from the plaques. Even more troubling, these lipid-laden microglia demonstrated a 40% reduction in their ability to clear amyloid beta compared to normal microglia from brains without disease. In essence, the very cells that should be eliminating the toxic protein deposits were becoming overwhelmed and dysfunctional, unable to perform their protective role. This discovery raised an urgent question. Why were these microglia becoming so heavily burdened with fat deposits, and how was this accumulation impairing their function? To answer these questions, Chopra's team employed specialized analytical techniques to examine the biochemical pathways active within microglia exposed to disease conditions. The metabolic cascade, leading to fat accumulation. The researchers discovered that microglia in contact with amyloid beta plaques and experiencing disease-related inflammation produce excessive amounts of free fatty acids. Under normal physiological conditions, microglia utilize these free fatty acids as an important energy source to power their activities, and a certain level of fatty acid production is not only normal but actually beneficial for cellular function. However, in the context of Alzheimer's disease, something goes seriously wrong with this metabolic process. Rather than burning these free fatty acids for energy as they normally would, the microglia closest to amyloid beta plaques begin converting them into triacylglycerol, which is a stored form of fat. This conversion occurs in such enormous quantities that the cells become literally overloaded with fat deposits, to the point where they can no longer function properly or even move effectively through brain tissue. The formation and accumulation of these lipid droplets is not static, but depends on both the age of the individual and how far their Alzheimer's disease has progressed, with the problem becoming increasingly severe as the disease advances over time. By meticulously tracing the complex series of biochemical reactions that microglia use to convert free fatty acids into stored triacylglycerol, the research team was able to identify the specific step in this metabolic pathway where the problem originates. They focused their attention on the final step of the pathway and made a crucial discovery. An enzyme called DGAT2, which catalyzes this final conversion step, was present at abnormally high levels in diseased microglia. An unexpected twist in enzyme regulation. What made this finding particularly intriguing was an unexpected twist. The researchers naturally assumed that if DGAT2 enzyme levels were elevated, they would find correspondingly high levels of the DGAT2 gene being actively copied to produce the protein. However, when they examined gene expression levels, this was not the case at all. The gene was not being overproduced, which meant something else entirely was responsible for the enzyme accumulation. Through further investigation, the team discovered that the DGAT2 enzyme was accumulating not because it was being produced in greater quantities, but because it was not being broken down and cleared away as rapidly as it should be under normal circumstances. This persistence of the enzyme meant that it continued to drive the conversion of free fatty acids into stored fat, causing microglia to divert their lipid resources away from energy production and cellular maintenance and instead funnel them into ever-growing fat deposits. According to Chopra, this research definitively demonstrates that amyloid beta is directly responsible for triggering the fat accumulation that occurs inside microglia. The presence of the protein plaques sets off a cascade of metabolic changes that result in these immune cells becoming laden with lipid deposits. Once burdened with these fatty accumulations, the microglial cells become profoundly dysfunctional, losing their ability to clear away amyloid beta and essentially abandoning their protective duties at precisely the moment when the brain needs them most. From discovery to therapeutic intervention. While the researchers have not yet fully elucidated what causes the DGAT2 enzyme to persist rather than being degraded normally, they did not wait to begin exploring potential therapeutic interventions. The team tested two different molecular approaches. One compound that directly inhibits the function of DGAT2, preventing it from catalyzing the conversion of fatty acids to stored fat, and another that promotes the degradation and removal of the DGAT2 enzyme itself. The results from these experimental interventions were remarkably encouraging. Promoting the degradation of the DGAT2 enzyme proved particularly beneficial across multiple measures of brain health. In animal models of Alzheimer's disease, this approach reduced fat accumulation in brain tissue, restored the functional capabilities of microglia, improved their ability to phagocytose and clear amyloid beta plaques, and enhanced various markers associated with neuronal health and survival. As Chopra explains, when the research team targeted the fat-producing enzyme and either removed it or accelerated its degradation, they were able to restore the microglia's fundamental ability to combat disease and maintain the delicate balance necessary for brain health. In essence, they were returning these immune cells to their intended purpose and function. Implications for drug development and treatment strategies. This work carries profound implications for how we approach the development of Alzheimer's treatments. For decades, pharmaceutical research has been dominated by efforts to directly target the primary pathological hallmarks of the disease, namely the amyloid beta plaques and tau tangles. Despite enormous investments of time, money, and intellectual effort, this approach has yielded disappointing results, with most drug candidates failing to provide meaningful clinical benefits to patients. Chopra argues that this outcome should not be surprising. In his view, directly targeting plaques and tangles alone will never solve the underlying problem because it fails to address the fundamental dysfunction of the brain's immune system. Instead, he advocates for therapeutic strategies that restore the function of immune cells like microglia, enabling them to perform their natural protective roles. The key to this restoration, according to Chopra's research, lies in reducing the accumulation of fat in the diseased brain. When immune cells become overloaded with lipid deposits, they lose their ability to maintain the careful balance necessary for brain health. By targeting the metabolic pathways that lead to fat accumulation, researchers may be able to restore the capacity of microglia and other immune cells to fight disease effectively and keep the brain in its proper equilibrium. The broader context of lipid biology and brain disease. This research is part of a broader renaissance in our understanding of lipid biology's role in neurological health and disease. Chopra serves as director of the Merck Purdue Center and holds memberships in several key research institutes at Purdue, including the Institute for Integrative Neuroscience, the Institute for Drug Discovery, the Institute of Inflammation, Immunology and Infectious Disease, and the Regan Streif Center for Healthcare Engineering. His work also contributes to Purdue's Presidential One Health initiative, which takes an integrated approach to studying human, animal, and plant health. The collaborative nature of this research, involving partnerships with Cleveland Clinic and led in part by Dimitrios Davalos, assistant professor of molecular medicine, demonstrates the increasingly interdisciplinary nature of modern biomedical research. Solving complex problems like Alzheimer's disease requires expertise spanning chemistry, immunology, neuroscience, and computational biology, among other fields. Looking forward, a new therapeutic paradigm. As the research team continues their work, they are opening up entirely new avenues for therapeutic intervention. Priya Prakash, one of the study's first co-authors, emphasizes the excitement of revealing how a toxic protein plaque directly influences lipid formation and metabolism in microglial cells. While much recent research in Alzheimer's has focused on the genetic underpinnings of the disease, this work paves the way for understanding how lipid pathways within the brain's immune cells can be targeted to restore function and combat disease. Palak Manchanda, the other first co-author, highlights the revolutionary nature of connecting fat metabolism to immune dysfunction in Alzheimer's disease. By identifying the lipid burden and the DGAT2 enzyme as a critical metabolic switch, the research reveals a completely new therapeutic angle. Restore microglial metabolism, and you may restore the brain's own defense mechanisms against disease. This shift in perspective from attacking plaques to restoring cellular function represents more than just a new treatment strategy. It represents a fundamental reconceptualization of what drives Alzheimer's disease at the molecular level. Rather than viewing the protein deposits as the primary culprits, this lipid-centric model suggests they may be more accurately understood as triggers that set off a cascade of metabolic dysfunction in the brain's immune cells, ultimately leading to the cognitive decline and neuronal death that characterize this devastating disease. As we move forward, this research offers genuine hope for developing more effective treatments that address root causes rather than symptoms, potentially offering millions of patients and their families a brighter future in the fight against Alzheimer's disease.